We're so happy to have Sarah Carey with us. We're so happy that we have a DJ. We're going to party after presentation. Sarah Carey, il a fait ses études de Floride, en Britannique, et ensuite il était postdoc dans United States Department of Agriculture, National Institute of Food and Agriculture. Et ensuite, maintenant il est scientifique senior dans National Institute for Biotechnology. Et évidemment, un travail sur cannabis, ça nous intéresse. Je vais parler par rapport aux chromosomes de sexe. C'était super intéressant. So, stage is yours.
Hi, everyone. My name is Sarah Carey, and I am a senior scientist at the Hudson Alpha Institute for Biotechnology. And I first want to thank you very, very much for the invitation to come and speak here today and the warm welcome. It's been really, really a wonderful trip so far, and I'm looking forward to getting to hopefully chat with more folks over the day. Today, I'm going to tell you about some really exciting work that we've been doing on the evolution of sex chromosomes in the Cannabaceae family. And so what I'm going to tell you about is my absolute favorite trait in plants and in biology in general, and that is the evolution of separate sexes. In the plant world, we call that the evolution of dioecious. It's a different term in animal world, but what we mean when we say dioecious, and I'm going to say it a lot, is there are plants that make separate male and female plants. They're separate individuals. And in the context of the flower itself, what we mean is we kind of consider this perfect flower, and there can be whorls of sepals, you have your whorls of petals, you have your carpels and your stamens. So your flowers can not always, but typically can self-pollinate and set seed. With dioecious flowers, females and males can both make sepals, they can make petals, but only your females will make functional carpels that will produce seeds, and only your males are going to make functional stamens that produce your pollen. So completely separate sexes, analogous to what we see in a lot of mammalian and animal systems. And I'm really, really passionate about this particular trait because it's pretty rare in flowering plants. It's in 5 to 10% of species, but has evolved, we think, thousands of independent times. And got the opportunity to see the wonderful greenhouses with cannabis. So if you're familiar with cannabis, you know that's a dioecious species as well. As we were walking in, I saw sumac trees and mulberry trees. If you didn't know that, those two species are also dioecious. So the plant that's outside that has the bright red fruits, that is a female sumac. Pretty fun. And so what we're really interested in is discovering what genes are associated with this transition from being able to have a perfect flower that self-pollinates and can set seed to having completely separate sexes, where now you're an obligate outcrosser. What genes in the carpel and stamen pathways have broken in order to have fully functioning separate sexes? And the evolution of dioecious also typically comes with the evolution of sex chromosomes. And so kind of the nice thing is, if we can find our sex chromosomes, we can narrow in on those carpel and stamen sterility genes, those dioecious genes. And so what we work on is trying to build fully phased sex chromosome references and leveraging those in order to identify our sex-determining genes. And what we do, so we want to build genome references that have all of our autosomes completely assembled, but specifically for these questions, we want fully contiguously assembled X chromosomes and our fully contiguously assembled Y chromosomes. And that's because we want to look in a very particular region of the Y chromosome, that very special region that does not recombine with its X counterpart, the non-recombining region, or SDR is something I'll probably call it a lot today. Because within that SDR, or our sex-determining region, should be our sex-determining genes, so our carpel and stamen genes. But we need fully resolved sex chromosome assemblies to do this. And so what I'm going to tell you about today are challenges and some of the methodological approaches that we've been using for and developing for studying sex chromosome pairs, and then tell you some stories about using comparative genomic analyses in the Cannabaceae family to understand their sex chromosome evolution, and hopefully get towards understanding the genes that are involved with sex determination in this clade. So first, the challenges and our ways of trying to tackle some of them. Sex chromosomes are notoriously difficult parts of genomes. They evolve very wildly, very rapidly, and very different than the autosomes. And if you, I didn't hit the one button. If you've been following the Human Genome Project as they were releasing all of the T to T or the telomere to telomere assemblies, you may know that the Y chromosome was the very last one to be released. And it's because of its complexity. It's a super difficult chromosome. And we'll focus here on this X chromosome. The X, kind of similar to autosomes, it's very gene rich. It recombines with other X's. It can recombine in the pseudoautosomal regions with the Y, but those are very small parts of the Y chromosome that can recombine. This non-recombining region, though, of the Y chromosome has evolutionary consequences. Over the 130 to 150 million years this Y chromosome has existed, most of the gene content has been lost. And instead, what fills most of this region are ampliconic repeats, super heterochromatic regions. All of these things, being gene poor, super repetitive, make it really difficult to phase and build these sex chromosome references until recently. But that's been a challenge, right? So getting a fully contiguously assembled Y chromosome has been tough. Now, if all sex chromosomes are like humans, we would now have some a priori assumptions. We'd know what we were getting into. But not all sex chromosome pairs are like the human XY. They're super, super diverse, and we typically don't know what they look like until we start diving in. And so one example I have here to show is the size of that non-recombining region where we are trying to find our sex-determining genes. That can be hyper variable. So asparagus is an example of one where the non-recombining region is 1% of the Y chromosome. Super small, kind of easier to narrow in on those candidate genes. But we have examples all the way up in bryophyte world, where fire mosses, the sex chromosome non-recombining region is most of the sex chromosome. Nearly 100% of it doesn't recombine. So now we're looking at 3,400 genes as our candidate. Pretty big. So variation just in that region, we're trying to find our sex-determining genes. It's also not always cytologically obvious which chromosome might be your sex chromosome. So we got lucky in things like hop and other species where the Y chromosome is smaller than the X. So if you look at male meioses, you could observe that this chromosome paired with the X of meiosis, but only then was ever found in males. So putting that together, you can identify, well, that's the sex chromosome pair. This is only ever inherited in males based on that smaller size. But most plants, instead, they're homomorphic. Try and figure out which of these is the sex chromosome umbrella. You can't just based on those kinds of patterns of smaller chromosomes and their inheritance alone. So really challenging to identify with just cytotypes alone. There's even more structural variation that can occur. So we just talked about how there can be homomorphic, where there's same size X and Y chromosomes, so hard to find cytologically. They're heteromorphic, where in the case like hops and the human XY, the Y chromosome is smaller. But there's other flavors of heteromorphy, like in cannabis, where the Y chromosome is actually bigger than the X. It gets more complicated from there. You can lose your Y chromosome and have a dosage or aneuploid system. So we'd be looking for a Y chromosome that's not there. You can have multiple pairs of sex chromosomes evolve, and multiple pairs of sex chromosomes that can evolve through autosomes fusing to your sex chromosome, your extant sex chromosome pair. So these are called neo-sex chromosomes. That fusion event is what is now defined as that neo region. So you have your ancestral part, and this part is now your newly incorporated sex chromosome sequence. So incredible variation just in the structure alone. Gets more complicated from there too. Which sex has the sex-specific chromosome can also differ. So we've really been focusing on XY chromosomes here, where that Y chromosome is only inherited through phenotypic males, or typically through phenotypic males. And so the sex-specific region typically goes through males. But the opposite can be true. We can have things like ZW systems, where the female now has the sex-specific chromosome that has that non-recombining region and the sex-determining genes. And we don't know this unless we start diving in. And if you find it with a cytotype, that's great, but often with plants, like I mentioned, homomorphic. So we just don't know which it is. Is it the XY or the ZW system? And that's important when you want to build a genome reference if we're after our sex determining genes, because we need to make our reference based on which sex is the heterogametic one. And so this is something that we've been working on, is tackling some of these challenges by an approach that we've been calling sex chromosome cytogenetics by sequencing. And it's kind of inspired by Nettie Stevens, who is the person who very elegantly first discovered sex chromosomes. She did this in mealworms. It was published in 1905. And she did this through the cytology. She found that pattern of the smaller chromosome being inherited exclusively by males and figured out there was a chromosomal basis to sex determination. And so we're taking inspiration from her and doing this approach instead with sequencing technologies. And so 3 of the things that we are doing with our cytogenetics by sequencing pipeline is first figuring out that first question of, is it an XY or a ZW sex chromosome system, so that we can make our genome reference be the heterogametic sex individual. And we do this with Illumina shotgun sequencing of known sex individuals. And I'll kind of get into a little bit how we do it, but trying to figure out how we can do this as cost efficiently as we can and with as minimal data as we can, but really get after that question. Is it XY or is it ZW? Once we have that, though, and we build our genome references, we can use CBS to phase our XY or ZW into a single haplotype. It's really important. We need our X contiguously assembled and our Y contiguously assembled into a single haplotype so we can make these inferences of what are those sex-determining genes, make our reference as useful as we possibly can. And then the third thing we can do is use these same data that we've been talking about here in order to identify that boundary of non-recombination. Where is that non-recombining region, the SDR, and where does it stop relative to the regions that do recombine? Because within there is where we expect our sex-determining genes should be. And so what we've been building is really based off of using sex-specific k-mers. CBS, or cytogenetics by sequencing, is really reliant on this pipeline using sex-specific k-mers. And what we do is we use our whole genome shotgun data of known sex individuals to find k-mers that are exclusive to one sex. So in an XY system, because the Y chromosome is only inherited by males, autosomes are inherited by both sexes, the X chromosomes are, the pars typically freely recombine, the only sex-specific sequence should be that non-recombining region. So if we find k-mers that are exclusive to males, they should come from this non-recombining region. And in a system where we don't know what the sex system is, if we find more male-specific k-mers, it should indicate that it's an XY system. The opposite then being true with ZW. If we find more female-specific k-mers, they should be deriving from that non-recombining region of the W. And if we don't know the sex system again, if we get more female k-mers, it indicates that we have a ZW system on our hands. And so we've been using this on dozens and dozens of species now. And we've been making really, I think, great progress in finding new sex chromosomes that we didn't know existed before. And so what I have plotted here is some of our output from the cytogenetics by sequencing pipeline. This is the proportion of k-mer sex bias. So the positive axis indicates that we found more male-specific k-mers, because it's not perfect. You'll find more male k-mers, but there's still noise genomically. You will still, in an XY system, get some female-specific k-mers coming out, which is an interesting thing I'm really interested in diving into where those are coming from, if it's maybe related to some other phenomenon. But we're looking for just an increased relative, more male k-mers. And if it's in the negative axis here, we found a ZW system. There's more female-specific k-mers. And these salmon circles are known systems, showing you that this pipeline is working. We knew these were XYs or ZWs, but these triangles are brand new sex chromosome pairs that we've discovered. And so it's not just XYs. We are actually finding quite a few ZWs as we start diving in, which is pretty cool. We've also been making great strides with being able to build our fully phased genome references, and then delimiting our par-SDR boundaries. And this is an example that we just published last week on Amberella trichopoda. And we used our CBS pipeline in order to identify female-specific k-mers because it's a ZW system. Pull all of those contigs from our genome reference into a single haplotype, so we fully phased it. and to a haplotype 1 here, and then use those k-mers to delineate this boundary of the non-recombining region, which let us examine this region and this really important plant. If you're not familiar with Amberella, it is the extant sister species to the rest of flowering plants. Really, really critical phylogenetic position in flowering plant world. So we were able to kind of look at this region of the sex chromosome at a new and kind of unprecedented scale and start tackling some questions like, what are the sex determining genes? I'm not going to dive too far into Amberella, though, even though I'm super excited about it, because I really want to tell you about Cannabaceae today. It's a really, really exciting group that we've been working on, and we've got some pretty nifty things that I'd love to share. And so those are some of the challenges. Sex chromosomes are pretty complex, but we've been working really, really hard to try and figure out ways that we can start tackling them and analyzing them. And so the rest of the talk, I really want to tell you about the Cannabaceae sex chromosome evolution and how we've been using our phased references that we've built in order to do comparative analyses in the Cannabaceae clade. And so if you aren't familiar with this particular group, we humans, we have a really, really deep history with cannabis/hemp and hops. We've been cultivating things like cannabis for, this is hops, but we've been for over 12,000 years is the number that I've seen reported. Really, really long time we've been domesticating and using cannabis for things like fiber and cannabinoid production and seed production. And hops also have a pretty long history with humans too. There's reports from over 1,200 years ago that hops have been used, the female flowers in particular, in beer brewing. So if you think of beer, you think of that bitter flavor and some of those fun, funky flavors in your brew. Those are coming from the flowers of the hop plant. And so we've been using these plants for quite some time. And importantly, though, females are the economically important plant. There are some uses for males, and I think particularly bast fiber in cannabis is sometimes males are used, but at least for kind of cannabinoid and seed and a lot of fiber and bitter acids, in the case of hops, it's females that are the more economically important sex. We also have a deep history with this clade in regards to their sex chromosomes. So we have been studying the cannabis sex chromosomes and the hop sex chromosomes for a century now. They are among some of the first pairs of sex chromosomes that were identified, not too long after Nettie, about 20 years later, but some of the first in flowering plants in particular. And here's the Vinga's paper, it was 1923, and Harada's paper in 1924, where these were first identified. I think it was the same year then as Silene and Rumex, if you're familiar with those. Those are also heteromorphic pairs. So a century we've been aware and studying these sex chromosomes, but we don't know a ton about them at the molecular level. And that's because of some of these complexities I've been talking about with building genome references on sex chromosomes. And so we have some linear expectations of how sex chromosomes evolve. Typically, they start off as an ancestral autosome, so they're freely recombining in a hermaphroditic population. Sex chromosomes then evolve, and I don't want to get too far into the different models and different theories of how that can happen, but let's just kind of jump to, we have one. And here we're going to focus on XYs because that's what we're going to talk about for the rest of the talk, is specifically XY systems. And what will happen is the sex determining gene or genes will evolve into a region of non-recombination so they can stay linked with one another and be inherited through the sex that they're beneficial for, in this case, for males. And what we're going to find is, because these are homologous genes and now they're not recombining, the genes are going to start evolving differently. They're going to pick up changes because they're no longer sharing their content. They're diverging. And we're going to measure that today with KS, which is our synonymous site substitution. So we're going to look for patterns of KS increasing over time for genes that are inside of this non-recombining region. And so here we have an early stage sex chromosome pair that's evolved. Over time, sex chromosomes start to take on their own linear kind of trajectory. They can pick up lots of repeats and become much, much bigger, as the case here. So they'll expand, like is what we see in cannabis. They're much, much bigger. They can then also capture more genes into their non-recombining region, so they can expand in that way, or they grab more things. And again, KS is going to be important because the first things that were captured are going to have much higher Ks, more protein divergence. But things that are more recently captured will have a lower Ks, so they haven't changed quite as much over time. And so start from an autosome, pick up a sex chromosome, things start to diverge where the Y can tend to get bigger, and we think that over more time, what will tend to happen is then genes will get lost. You'll get left with things that are just super important for fertility or sex determination, but most other stuff, if it's not super deleterious, can get lost. Your Y chromosome can shrink back down. Think of the human XY system, much, much smaller, not a lot of genes, mostly repeats. And so this is kind of the model that we've been operating on for a long time. We thought this is exactly the trajectory of how sex chromosomes evolve. Cannabaceae does not follow this model. It defies it in every possible way because nearly every flavor of structural variation that I showed you exists in the Cannabaceae sex chromosome pairs, but they evolved at the same time. So they share an ancestral origin. It was about 40 to 50 million years ago. Cannabaceae had an ancestral set of autosomes, but since their divergence, cannabis now has evolved a Y chromosome that's bigger than its X. Beer hops instead, same origin, the Y chromosome is smaller than the X. There are wild hops that grow in America, and I think probably Lupuloides is one that grows here. They have homomorphic, cytologically, homomorphic sex chromosomes. They're technically smaller, but if you looked at it under a microscope, probably couldn't tell that they were different from one another. So we call them homomorphic. There's also neosex chromosomes. Remember I said there's fusions of autosomes to the ancestral sex chromosome pair? That happens in Japanese hops. And there's also multiple systems and other things out there too. But this is kind of incredible. If we thought there was a linear trajectory and only time made these cytological variations, that really can't be true because we see all this variation from the same ancestral sex chromosome pair. So this is just a really exciting clade, in my opinion, to look at the molecular evolution of these regions. How are we getting such extreme variation from the same start point? But the thing that we're super after is, go back one more, is we want to build genome references. We want to look at all of these different sex chromosome cytotypes and uncover some fun patterns, but what we want to do is do comparative analyses in order to look back in time. We want to look and try and figure out what those ancestral sex determining genes are in things like cannabis and hops. And so to do that, we've been building our genome references. Here are 3 hops that we have done for XY males. And so what we've got are, here are high C contact maps for a variety Lupuloides, variety Neomexicanus, which is more in the southwest of the states, so more arid environments. And this one is our beer hop, so European hops. And these contact maps, what they really highlight is that we have 10 chromosomes, which is what we were expecting, fully and contiguously assembled in each of these. We do have it for both haplotypes. I just have one here kind of for simplicity, but the genomes are all coming out to the expected sizes we were hoping for. So hops have 2.5 to 3 gigabase size genomes. They're fairly large. But we had really nice big pieces that we were using to pull together. So nice references that we've been able to pull together, but importantly for the sex chromosomes in particular, we have been able to fully phase our Y chromosomes from our X chromosomes. And so what we have here are, those are our male-specific Kmers, our Ymers, highlighting that non-recombining region of the Ys. And so here's the Ymer the SDR in Lupuloides, Neomexicanus, and beer hop. And you can see that there's a little bit of noise, and that's actually, I think, some interesting biology happening that we could talk about later. But what's really cool is the size of the chromosomes. So this one's 244 megabases, this one's 245. That's about the size of the X chromosome. So remember I said these ones are homomorphic. They're about the same size. So we recovered that in our assemblies. And then the beer hop is much smaller. It is, in fact, much smaller than its X chromosome counterpart. So we got the sizes we were looking for. And these regions of the non-recombining region are about 80% of the overall chromosome. Huge. And also meet cytological expectations. So there's some nice, beautiful cytological work that showed that most of the Y chromosomes in hop and hemp don't recombine. So this is where we're going to be looking for candidate genes, is unfortunately most of the chromosomes. Similar things in cannabis. So we have XY references now for 2 cultivars of cannabis. We got our expected 10 chromosomes in both haplotypes. Expected chromosome links, these are the numbers straight out of the assembly. They drop a little bit once you start kind of filtering things, but nice big pieces that we were pulling together. And again, importantly, our sex chromosomes are the sizes we were expecting, 110 to 115 megabases, and they are larger than their X counterpart, which is what we were expecting. And the non-recombining region is about 70% of these chromosomes. So also meets cytological work. So huge parts of those SDRs. And this was pretty complicated. When you build these references, the assembler doesn't always know anything, like where things are supposed to go, unless you can give it parental information. It doesn't know an X from a Y. It doesn't know paternal versus maternal autosomes. And if you can't get trio binning, which is what that's called when you use your parental information, what do you do with something like a sex chromosome? Something like this that's so very different from its counterpart. You have to go back and you have to find it. And that's what we did with CPS, is we found the Y chromosomes using these male-specific hamers, pulled them into one haplotype, and then we're able to pull them into scaffolds. So fair amount of manual curation, but we got there. And so now that we have these references, we can start really diving into the fun biological questions. The first one, of course, was, all right, so when did they evolve? We knew there was a shared origin based on some wonderful work from Printout, but now that we have Y chromosome references, which are new for hops, and some of them are new for hemp. I know we had some, but not maybe as fully phased with for them. Now that we have them, how old are they exactly? And so what we've done is a couple of different things. I'll first point us over to this side of the tree. So this is a phylogeny that was built off of 775 single copy loci across hops and other parts of Cannabaceae, Moraceae, and other species in the Rosales. And Phil Bence is the one who did this great work. He built this phylogeny and then used plastomes in order to date the phylogeny. And so what we have are the mean ages for particular nodes on here. And this is our node for cannabis from the split to humulus. So about 36, 37 million years, although there's a pretty wide confidence interval, is where we were thinking we were going to see this evolution. And using that KS, remember I said the oldest genes to stop recombining will have the highest KS values. We're able to kind of validate that we think it's about 47 million years old. And that firmly plants it on here. You don't see the confidence interval shown here, but it firmly plants our STR evolution right there, just before cannabis and humulus, which is an important and interesting finding because mulberries, like your mulberry trees outside, and figs are in the sister family Moraceae, so sister to Cannabaceae, and they're also dioecious, and they also have XY chromosomes. So my question was, are they the same evolution? And so this date says no. They are very different from one another because the divergence between Moraceae and Cannabaceae much greater than this date that we were able to establish using the genes in the sex chromosomes. The other thing then was we used synteny just to look and see, are they the same chromosomes in another way? And that's what this particular side of the panel is showing here. So these are synthetic relationships between the hops that we've assembled. And the gray lines identify the ones that we were just talking about. And there's a few other varieties that we have in this tree. But the gray is collinear gene order. So within hops. And then we switch to within cannabis down here. And then we switch to the Moraceae family down here. And in green, I've highlighted the sex chromosomes in the Cannabaceae clade. So we have our nice kind of shared origin. And it's a little complicated just because hops and cannabis have their genomes numbered differently. So kind of have to track it a little bit, but they are indeed the same sex chromosome pair, which is what we were expecting. But mulberries, so this is Morus notabilis down here at the bottom. This is the SDR, is that tiny little piece right there. And you can see in purple, that's a completely different chromosome from in cannabis and hops. And the same thing in ficus. So figs have a completely different chromosome from cannabis and hops, but also from mulberries. So we have 3 different evolutions of sex chromosomes here, which we've additionally validated with gene trees. There's nothing in these non-recombining regions that's shared between the mulberries, figs, with each other or with cannabis. So pretty confident that this is a sex chromosome that evolved just prior to the divergence between cannabis and hops, which is still really cool. We didn't draw back any further in the nodes, but this is, as far as I know, the oldest pair of chromosomes, sex chromosomes in flowering plants. I say as far as I know because once we start diving into the thousands of other evolutions, we might find some different patterns, but at the moment, that's pretty old and the oldest that we know of. Most other pairs we've looked at, much younger, which tracks with possibly why they're so homomorphic. And these ones are so variable in their cytotypes. Linear trajectory might not follow exactly, but over a lot of time, have lots of time to start to evolve these really different cytotypes on the sex chromosomes. So hops and hemp share an X chromosome pair. Super important and helps us then be able to kind of still walk back and see if we can find those sex determining genes. With having a shared sex chromosome pair, there's still been a substantial amount of change that's really important to the XYs that's occurred. And so what we have here are our gene and repeat motifs and our syntenic relationships between just one hemp assembly and the X and Y included, and one hop assembly with the X and the Y. And these stars indicate our centromeres. And so our genes are in orange and yellow, repeats in blue, centromeres, and then synteny. And Laramia Kozbeck is a wonderful PhD student who did the centromere work, which is going to be super helpful here in a second. And John Lovell wrote this program, GeneSpace, and actually modified it so that I can show you this relationship today. Because hop genomes are 3 times bigger than hemp, we had to scale hop in order to make this kind of relationship actually visible before it was wild. So really grateful for their help with this. But the really important thing I want to show you is here's our cannabis XY pair. And here's the syntenic relationship between the extant hop sex chromosomes. And you can see there's another region on those the cannabis XYs. There's a fusion to the cannabis XY pairs. Cannabis XYs technically have neo-sex chromosomes, which is something that we didn't know until we compared them to hops. They looked like standard XYs, but when you have this kind of frame of reference of other individuals in your group, you can see these kind of genomic changes that are super important for the structure of your genome. And so what appears to have happened is there was a centric fission, so that stars our centromere on this chromosome 1 here, and then that fused to the XYs and hemp, potentially driving some of that size variation. They are a bit bigger in relative size compared to the hop. So interesting things already happening. We've got some major structural variation. There's a few other things that are happening in here too that are a little harder to see perhaps, but the X chromosomes and hops have an inverted translocation where you can see that the genic region that typically is more towards the telomeres is more interstitial. It's closer to the centromere, which if you're a hop cytogeneticist, you might recognize that pattern. The X chromosomes had their telomeric motifs more towards the centromeres on the X chromosome, which made it handy for tracking them, but kind of a curiosity of why? That's why. There was a nice inverted translocation that drove that pattern. But kind of focusing in within a clade, we're going to look at the hop Y chromosomes now. There's a ton of structural variation that happens in the chromosomes. We've been focusing on genes and repeat motifs, but the overall order of the chromosomes is also hyper variable in Y chromosomes. And so we can have our genes and our repeats here and our syntenic relationships, or in some cases, lack thereof. And first, we want to focus here on these 2 wild hops, so Neomexicanus and Lupuloides. These are sympatric varieties, so they have overlapping distributions in some parts of the states. And we clocked these as having diverged about 200,000 years ago. So just giving you some context for the amount of time variation between these 2 sympatric varieties. This is our SDR. I know the Weimers are a little tough to see because the Y chromosomes are mostly repeats. But this is our non-recombining region right here. And this is our PAR. This is the part that technically freely recombines, although with these SVs, maybe not entirely. And what you can see is right when we hit that SDR region, yeah, repeats pick up very sharply right at that line, and we become very, very, very gene poor. There are very few genes in this non-recombining region. So a little stressed, and I was like, most of this region is the SDR. Oh, we're going to have some trouble finding these genes, aren't we? But it turns out there's not that many in there. There's only 300 or so genes in each of these SDRs, despite how big these chromosomes are. So convenient. But the really, really fascinating thing to me in this plot is just look how much structural variation has occurred. Look at how quickly things have completely rearranged in this non-recombining region. It's hyper variable, hyper cool parts of the genome. And as we move into looking at Beer Hup, which again, this was our Y chromosome that's smaller, you can see that variation. These are true to size. That Y in Beer Hup is much smaller, in fact, but it has almost no synteny left with the wild hops, almost none. And it's about 1.6 million years diverged from wild hops. And one of the most fascinating things to me about this is we cross these varieties. Beer Hup is mostly lupulus, but we cross it with Neomexicanus and lupuloides, and how? Now, the Y chromosomes aren't going to cross with one another, but when you look at this variation, and there's a lot of variation on the X chromosomes too, it could explain some differences and challenges associated with crosses of distinct varieties if you've encountered them. But this region, still, yes, gene poor. We're after our sex-determining genes after all. So we're going to keep kind of doing more comparative analyses. And what we're going to do is compare our X to our Beer Hup, to our Y to our Beer Hup, to our X chromosome. And we're going to add in some more Cannabaceae comparisons because want to find those sex-determining genes, what's in these Y chromosomes that are shared across all these evolutions that have been there since they evolved. And so what this is exactly that. This is an X chromosome in Beer Hup. You can kind of see that interstitial kind of translocation now. Here's our genic region is more inward, which is very fascinating. Here's our Y chromosome. And our syntenic relationships between the X and the Y also not really there, except for in the recombining region. And here's our KS. It's come back. And what's really interesting is KS is pretty consistent across the entirety of the non-recombining region. Remember, I kind of talked about there being a stepwise addition that can happen, where the first captures will have higher KS values, more captures will have more reduced KS values. Most of the non-recombining region was captured at the same time, both in hops and in cannabis. So most of it. Maybe one event, like an inversion or something, but we can't quite figure that out because they're not in the same order anymore. But this still doesn't quite narrow us in on exactly where we should be looking because we are trying to find where the highest KS, and then we have the drop-offs, but still kind of the whole chromosome, except for the PARs. There are some independent captures along the sides, a little tough to see. There's a few of them in Beer Hup. Mostly cannabis has a bunch of independent captures, which is really fascinating. But when we start to do more orthology-based analyses, taking those gene models that are low in the Y in our hops and then comparing them to cannabis, how many genes are shared across all Ys? So that can help us look back in time. And that narrows our list down to quite a few more. There's only 54 genes that we've been able to annotate that are on the cannabis and hop Y chromosomes. And what we wanted to do is then overlap that with what are known floral pathway genes and ones that fit the development of hops and hemp. And there's only 5 floral pathway genes we identified. There's not too many, but none of them really fit the development of hops and hemp. None of them are obvious sex-determining genes. What there are instead, though, are really interesting pollen fertility genes. And so we have REN4 and ATSPP. They are involved with pollen development and pollen germination. And again, they are shared across both cannabis and hop Y chromosomes. So feels highly probable they have some really important functions for the development of pollen in both species. But nothing so far that is an obvious sex-determining gene on this Y. So what that kind of points to is this actually not on the Y chromosome? Is it actually something else that I didn't tell you about yet, which is an X autosome balance system? And what that means is the inheritance of one X chromosome would develop a male, and the inheritance of two would develop a female. That's been posited for this clade for quite some time, and it was based off of some really elegant crosses with polyploid sex chromosome pairs, and really strongly showed that actually it's really just how many X chromosomes you have. And I saw that and I was like, I want to look at the Y anyway. I think something's going to be there, but so far nothing obvious is popping out. So could be an X autosome balance system. We've also compared these results, and I'm not going to show you any figures on it or anything, but we have monoecious cannabis assemblies that we've assembled, that we've assembled too. And with Monaceae, these individuals now produce both male and female flowers on the same plant. So instead of being on separate individuals, they're on separate inflorescences, but same plant. And nothing pops out there either. So the theory we thought we were gonna find is, now that we have Y chromosomes, we know what genes are supposed to be there, we know what the Y looks like, we have Y-specific sequence. These monoecious lines may be something from the Y chromosome translocated to the X in these individuals, and that's what's causing Monaceae, because they seemed XX in their karyotype based on C values and genome size estimates. But there is nothing from the Y on these monoecious lines. No Kamers mapping, no gene trees. I looked at 1,500 gene trees. Nothing appears to have moved from the Y chromosome. So pretty strongly starts to fit this X autosome balance system as well. So don't have a candidate gene there yet, because that's going to take, I think, quite a bit more work in order to really narrow in on that. But I think we've potentially started to rule out in a lot of different ways it's actually not the Y chromosome, which really, really defies the whole model that I've been showing you. But extremely cool. Just another way that sex chromosomes can evolve. And so, much with this here, but there are other interesting genes on the XYs. So we can't find that gene just yet, and there are some pretty interesting flower genes on the pars too. But what I want to tell you about is beer. And specifically the genes associated with making bitterness in hop cones and what's used then in bittering beer. So just a little bit on the background of hop cones. So the female inflorescences produce these structures called lupulin glands. And so they're kind of these orangish regions that are nested up inside of here, inside of the strobilus. And what these lupulin glands produce are alpha and beta acids. So these are what determine bitterness in beer. These flowers also provide those essential oils, which give you that fun flavor, think your tropical notes and other things that you get when you have like IPAs and other fun beers. They give you that aroma, they give you that taste. But we're going to focus on bitterness. And so there are some really, really wonderful works that have identified some of the genes associated with bitter acid production. And there are 3 key enzymes that are really, really important. There's lots of enzymes, but these ones are really, really important. So VPS, which is valeraphenone synthase, sorry. And then PT1 and PT2 are prenyltransferases. These 2 genes are super exciting to me because they are really important for bitter acid and hops, but they also are really important for cannabinoid production in cannabis. So they are the same gene family. They do a lot of different things. So they help with the bitter acid prenylation. They also help with xanthohumol. They're really important. If you don't have PT1 and PT2 functioning, you don't get bitter acids. You could probably guess where this is going to go in the next slide. They're on the X chromosomes. So these really, really critical bitter acid production genes, this production that occurs principally in the females of these species, these genes are on the sex chromosomes. Curiously, they are actually in the par, so they're in that region that's typically supposed to recombine, but in our beer hops, so this is actually a USDA cultivar that is in their breeding program, there are 2 inversions and a translocation jutted right up against that SDR. So this region that's supposed to recombine, most of it actually doesn't. It's not sex-linked necessarily, not in this individual. This is where some of those kind of interesting peaks were when I was showing you that plot. that could be on its way there, but as far as our evidence shows, this is not sex-linked in our beer hop. But right here are PT1 and PT2. They're tandem duplications located right next to one another. They are also just downstream from FT, which is also locked in the same inversion, which is pretty interesting, especially thinking about the breeding angle if you're trying to select for a certain parental transferase in a certain flower time. Stuck in an inversion. And we've looked at other previous QTL analyses for bitter acid and been able to use sex-linked markers that have been identified in order to track their QTL hits back to the sex chromosome. So there are other parts of the genome that get hit for bitter acid production, but a key one that shows up are the sex chromosomes, which is consistent with the parental transferases being there. And so this is really cool. And that's why we feel pretty confident in saying that sex chromosomes made beer. So next time you think about a beer, think about sex chromosomes. They contain our bitter acid genes. So really, really key genes are on there, but also somewhere they have those sex determining genes that decide or dictate whether you're going to have a female that produces those wonderful cones we use in brewing, or whether you have a pollen producing individual. And so sex chromosomes made beer, they have these genes. And kind of just to summarize this section up, we have Y assemblies now for hops and hemp. This was a really big challenge for a long, long time, but we have them. So now we can start diving into these kinds of really cool questions. They're hyper variable in their structure. And one thing I want you to take away from that is from here going forward, we're making these claims based on a handful of genome references, kind of how big the structural variant landscape is and all these kinds of things, but really one genome reference isn't sufficient. And we need to start thinking about building pangenomes. And I don't know how we're going to do that in a Y chromosome that doesn't align. That's my next challenge. But one reference isn't enough. You really need multiple individuals in order to understand the landscape of your sex chromosomes and autosomes, genome-wide. We have some interesting pollen genes that are shared on the Cannabaceae XYs, so potential candidates for those kinds of questions, but no clear Y-linked sex determining gene. So possibly is actually an X autosome balance system. Seems to fit that. And then, yeah, our bitter acid genes and parental transferases in cannabis as well are on these sex chromosomes. And so really the major takeaway I really hope that you get from this is sex chromosomes are complicated. I heard there was a talk about sex chromosomes yesterday too. And so they are complicated, but they are so worth investigating because I wouldn't have figured out or found this pattern of these bitter acid, these really critical genes to the phenotype of this plant of study unless we had references like this. So with that, I would like to thank all of my collaborators, my was postdoc advisor, but now I guess mentor, my boss, Alex Harkis. Laramia Kozbeck for all the incredible work she's done with all the repeat analyses and the centromeres. John Lovell's modifications to gene space to be able to make these plots. And Philip Bentz for his work in the phylogeny. Haley Hale, Hannah Mueller, and Zach Maharg are our molecular team who did all of the extractions for all of the work I presented today. I just sit at a laptop and do all this work. And then Josh Havel and Gary Muehlbauer, who are the hop experts who provided all of the hop tissue that I was able to present today. And then lots of other collaborators on the cannabis side of things. So Nolan Kane, Todd Michaels' group, and I don't have it up there, I should have it, Rafael Guerrero also on the cannabis side of things because I thought that I wouldn't have the cannabis data that I was able to show you today. And also thanks to our funding, so the USDA postdoc fellowship that I had to study the hop sex chromosomes, Alex's USDA NIFA to study hemp sex chromosomes, and his career grant to study sex chromosomes in general. And then thank you for your time.